mitosis and meiosis, like color-coded like both of them, and it kind of takes a few minutes. So this is tedious, but then I'll label things that'll like help on your test, okay? So I'm gonna start. Okay, that's this. I'm starting with G1S and G2E. These phases are known as interphase, okay? Interphase is kind of, we look at it as sort of a resting period, but really that's mostly G1. You're never going to do the S phase, like a cell won't go through the S phase unless it's planning to divide anyway, because otherwise it's like too much energy, it doesn't want to waste its time. But this whole period is before we've started mitosis or meiosis, okay? So this whole thing is like resting phase. G1, we're just kind of growing a little bit. But during S means synthesis, so just to clarify, and I'm going to kind of draw it for you. During the synthesis phase, each chromosome copies itself. I know we normally draw it as an X, but what I'm trying to remind you of, like instead of it, I'm going to start drawing them as an X just to go faster. But I'm going to first point out it's going to be a chromosome, and then exactly the same identical chromosome next to it. Okay, so all it's done is copied itself. It started here, and it copied itself. The other one's going to do the same thing during the S phase, okay? And they're just hanging out. In reality, the DNA is totally unwound, and you can't even see it anyway. But we have to draw it that way, or else I can't really show you like how many chromosomes there are because it's invisible. Right? G2. I'm just going to add here. All right. It kind of looks the same. In G2, like there are things happening at the enzymatic level, like invisible level, I'm gonna say, but the chromosomes don't change right now. All right, are we good so far? We're good. All right, we're gonna go two different routes for the mitosis versus meiosis. So I'm gonna go up here versus down here. This one will be mitosis, and then this one will be meiosis. Okay. So each of them, during mitosis versus meiosis, it's somewhat similar, like not a lot happens, oh sorry, I don't know where it's here. Between here and here, what's really happening is, remember I just told you, these would actually be all uncoiled and you couldn't really see them very well, so this is where they coil up. So for, I'm going to just put the lids on the pens and not hold 60. Okay. That's smarter. Okay, so mitosis, these guys are just hanging out somewhere in the cell. It doesn't really look much different, right, than during the S and G2 phase. They're just still hanging out. Meiosis is a little bit special though, because at this phase, these chromosomes have gotten really close and they are kind of crossing over one another. So this is where crossing over happens. So here's your first hint. If you were given pictures, the chromosome, again, the chromosomes are the same. You have the same stuff in each cell, but it's really, really important that these guys are called tetrads. And they're homologous chromosomes. And crossing over is happening. Okay? So, again, the same stuff is in each cell, but this is where crossing over happens, all right? Also, to clarify, this is super confusing. I'll kind of connect them at the centromere. These are connected at the centromere, okay? The left side and the right side of each of these is identical. They've replicated, all right? One of these singularly is called a chromatid. Okay, a chromatid is 1/2 of a replicated chromosome. I'll tell you when it stops being called a chromatid though, so I'm just going to point it out. A chromatid would be this half of the paired chromosome, okay? Two chromosomes are hooked together at the centromere, or connected together at the centromere, to make a chromosome. So we would call this entire thing right now a chromosome. To confuse you, as if you're not already confused, This singular stick is also called a chromosome. Even though you're like, wait a minute, but you just told me that half of it, that's a chromatid. Only because it's attached to a partner. If it can travel on its own, if it can swim independently, it's called a chromosome. So that's a chromosome. This, though, also is a chromosome. This singular stick is now called a chromatid because it can't swim away by itself. That is not really the technical term. That is not the scientific way of defining it, but that's the only way that I can describe it without confusing myself, okay? Swimming independently, chromosome. Attached to someone, like a conjoined twin. I guess that's kind of strange to think about, but there you go. So, you know, that's a way to think about it. Hi. Thank you. I appreciate that. Okay. We good so far? All right. So that helps with the question on the test because it's confusing and the word, it's just the wording and it's stupid and I'm sorry, but there you go. So this is called prophase. This is also called prophase, but I'm gonna call it, but it's prophase 1. Right? So far, kind of simple. Your next steps, though, are both metaphase, obviously, but they look different. This is the one that I drew on the board today during class, for all the classes. So this is mitosis, right? During mitosis, the chromosomes are all gonna line up in the middle of the cell, but in a single file line, okay? Remember, there is no crossing over, there are no tetrads. All I want to do in mitosis is make an exact copy of what I already had. So I have them line up single file. In meiosis, I take these guys, which have now probably crossed over. So maybe this has a different colored leg, I don't know. This maybe switched legs with this guy. But during my metaphase, let me write that down. During metaphase 1, and then this is just regular metaphase, and yes, the number after it is important, it would be marked completely wrong. If you just wrote, this is metaphase, you're wrong. No half credit, okay? Because you're not telling, you're implying that it's mitosis versus meiosis, okay? So that's not correct. Oops, I'm erasing chromosomes, sorry. Okay. During metaphase 1 of meiosis, look, I have the same contents of the cell, right? A red one, a red one, a blue one, a blue one. But these guys are in a double line, and these guys are in a single file line. Cool? Okay, cool. Next, you know what comes next. So anaphase. And then anaphase. It's getting more and more messy. All right. During anaphase of mitosis, the chromosomes are going to get pulled apart, but they are already sliced down the middle, okay? So they don't look like X's anymore. In mitosis, they only look like X's temporarily, just for those two phases, like prophase and metaphase. And then if we were doing a lab with them, you'd have to slice them up the middle like you do, okay? In anaphase 1 of meiosis, this is important because this is where independent assortment happens. Okay? Because some of my chromosomes will go to the left, some to the right. Maybe both blues would go to the right. I don't know. But I'm going to draw it such that this one goes there and that one goes there. This one there, and then, oh yeah, and we switched a leg, so there was some crossing over, right? The reason we're saying it's independent assortment is because I've now mixed, like, my reds and blues, okay? So if this side, let me block. If just this side becomes a baby, and the red side maybe came from grandma, that baby is going to look more like grandma because it got more of grandma's genes, right? It's a bigger chromosome. Just got more of grandma's genes than grandpa's. If it's that side, that blue one, that blue chromosome, the larger one, came from grandpa maybe, okay? And so it would, that baby might look more like grandpa, okay? So we've now mixed things up. So we've already created we are creating two cells that look different than one another, okay? Next is telophase / cytokinesis, and then telocyto 1, same idea. But after I've split apart the cells in mitosis, I'm all done already, okay? I end up with one large, one small of each chromosome, okay? What you're supposed to notice of this Here's my one large, one small of each chromosome. That's literally exactly what I started out with in G1. Like, I went from here with one large, one small of each, and I made two cells that are exactly the same. Remember, there's no opportunity for crossing over, so I didn't jumble any of my DNA. The reason there's no crossing over is because they never lined up next to each other. They only do this in meiosis. And you're like, well, why do they only do it in meiosis? Because only the ovaries and the testes tell them to do that. There's special cells in the ovaries and testes that do this. There's literally no other cell in your body that'll do this, where they line up like close together. So there can't be any crossing over, which is good. Okay? Good. All right. Telocyte on this one, again, we're going to make two cells. And so I have my big red chromosome there. And then I had like a blue leg here. And I had a red leg there because of crossing over. Now I got to get into we have to go to meiosis too. So remember, mitosis is completed. Okay, so this is all done. We've already made what we needed to make. Oh, the other thing that I was going to say is right here. It's independent assortment and segregation. So remember, segregation means separation, and independent assortment meant like the reds and blues don't have to go to the same place. So both of Mendel's, like his big laws, happen primarily in anaphase 1, okay? His laws imply that if everything works perfectly and meiosis goes really, really well, then I'm gonna get cells that have different combinations of alleles, right? Like different reds and blues, but also that everybody ends up with the right number of chromosomes. So far so good, there's not been any problems, right? If we follow these, so we'll do, let's do cyto and prophase. Nothing looks any different in prophase 2, so I'm not going to draw it again because it uncoils and it recoils, but nothing changes for prophase 2. So telophase 1, cytokinesis 1, and prophase 2 all look kind of the same. But now we'll get to metaphase 2, all right? In metaphase 2, they're going to line up in the middle. And I know this is there, and this is there. Okay. Now, here, oh my gosh. Here's where we get confused, unless you're already confused. The confusing thing is if I gave you this cell, like right here, or this cell, you might say, how could I tell the difference? Like between, okay, because there's a lot of cells up here, I know. If I asked you, oh, never raise. Hey. Hey. I am, yeah, I gotta redo what we're doing. Yep. All right, thanks. Cool, okay, thanks, bye. Okay. If I gave you this cell versus this cell, you might be able to tell. You'd be like, well, they're all doubled up, and she said that they double up. Maybe you'll remember. I hope you remember. If they're in, like, groups of 4, then you can probably go, like, okay, that's meiosis, and it's meiosis 1 because they haven't separated yet. So you can probably tell the difference between these. Here's the difference between this one and this one. Obviously, it's the number of I mean, I know, there's more over there than over there. But if it's not that obvious, look, this one, I only have one big and one small, right? I have one of each type of chromosome, like a big and a small. This one, I have a big, a small, a small. So if you see mitosis metaphase, or metaphase of mitosis, it's a single file line, just like it is over here. But here, I've already kind of like eliminated some of like the duplicates, you know what I mean? Like I used to have 2 bigs and 2 smalls, and now it's still a single file line, but I only have 1 big, 1 small, and then I have another cell with 1 big, 1 small, okay? So if you only see one of each type, you know it has to be meiosis, okay? Because in mitosis, we always have one from mom and one from dad. Our last parts on here are going to be, oh, no. Anaphase 2, and then it'll make telophase. So during anaphase 2, you'll notice, I know you kind of could tell when I gave you the pictures today. Oh. No, that's not the right one. Okay, what am I doing? Okay. During anaphase 2, again, you could tell the difference because you only have bigs and smalls. This is also a place where a chromatid is now called a chromosome. Alright, so here, we'll say this thingy, this right there, is chromatid. As soon as it is isolated and it is swimming on its own, it's now called a chromosome. Didn't really change at all, but now it's called a chromosome. Okay? Now, the very last thing makes 4 cells, right? The 4 cells, oops, don't like that. 2, 3, and 4. What we know, oh yeah, come on.
So when it separates, so at first it's also chromosome, when it separates, I mean, there's another chromosome.
It's a chromosome and a chromosome. So when it's like that, okay? Each of my hands is a chromatid. Together, this whole thing is a chromosome. Now each of my hands is a chromosome. There are no chromatids. There's nothing called a chromatid anymore. Crackers or crappers?
Crackers.
Oh, that's better.
You're so funny, Derek.
Crappers is kind of icky.
I hate him so much.
Now,
So irritating. He's my super ex, okay? It's equal here.
alright. Here's what we ended up with. I now have 4 haploid cells, right? Haploid means half of what I had before. So, if we can compare, number 1, if we look back to the very, very beginning, which is a long time ago, the very, very beginning we had just at G1, this was our regular old cell hanging out doing nothing, okay? We had a big blue, a big red, a small blue, a small red. Now, I only have, how many times can I throw the time capsule? Like, is it going to happen every minute? I don't know. And I threw it into the broken glass container. Hold on. That was stupid. Okay, and it's dirty. That's
okay.
great. I know, it's fine. It's just broken glass. I can handle that. Okay. I started out with 1 big red, 1 big blue. Now, in each of these, I only have 1 big something, right? Here I have a big red, and oddly, it has a blue, like, stick on the end, right? It happens to be paired with a blue small, all right? So, first of all, we can see independent assortment because I don't have all reds in 1 egg or sperm. And we can see crossing over because I have some chromosomes that have different parts to them. Okay? Good on that. Alright. The final things, wait, I need to point out something else. I guess the thing that'll help you is, if you can remember, well, all of it'll help you. If you can remember where the X's don't look like X's anymore in each of these, then that's going to help you figure out, decipher the pictures I'm giving you. I know, let's go. Pizza?
That's gonna be
Ice cream?
That's our class
Not for
party,
you.
right?
Everybody else.
Wait, that's our.
How did you know?
Did you
Oh,
say-
excuse me.
Did you say that?
Yeah, I did.
Oh. Oh wait, did you ever figure out about that?
Did I have a Did I have a shaved head?
Wait, did he get DQ?
Oh, my alter ego.
Did he get DQ?
I never. I'm a failure. Hold on, I'm ready.
Okay.
No, I'm
We need to know that. I need
We
this
do
party.
need to know. Wait, but
My
you know, we have a
self-esteem
mixed
is-
crowd right now, so let's because.
You guys feel
I love all classes equally. And all classes are equally deserving except for not, because one class have the competition and the others don't, which is the rule, right?
all the other periods besides first period cheating.
Okay.
Honestly.
What was I saying?
I'll be back 'cause I got hurt for any damage.
That's all that.
Not the same school.
Maybe that's it.
No.
What'd you get?
Yeah.
What'd you get? 43.
You're 40.
Oh, that. I got 48.
Okay.
Whatever.
All right, pointing out two more things using your test. Please notice the difference between G1 and like G2, okay? It's kind of an easy, or I don't know, like if you can remember it, it's an easy difference. G1 is just 6 single chromosomes, one of each, right? G2 has, they're still called chromosomes, I know it's kind of annoying, but they are replicated chromosomes. So they look like X's, okay? So S stands for synthesis. That means that's when they're copying their DNA, all right? Also, these, you know, they are identical to each other and they're identical to the parent cell, okay? So very important that mitosis creates identical cells, all right? Yep, that's good. This whole thing, that's another important thing. The entire board is called the M phase. Okay, so literally the whole, well, mitosis, the top part of the board. Sometimes they say, or always, they say G1 phase and then G2 phase and S phase, okay? So all 3 of these phases are in interphase. Then they'll sometimes group all of this part together in the M phase, and it just stands for mitosis, okay? Then, there you go. That's pretty specific, that was good. You know that. Then the picture should be fine. Okay. Finally, there's a question. I think you already know it because I told you today in class, but meiosis is 2 divisions, mitosis is 1 division. Both have only 1 replication or duplication of chromosomes. So we replicate our chromosomes, divide once, so mitosis takes us back to normal. But replicate our chromosomes, divide twice, that's why it halves the number, or cuts in half the number of chromosomes that we originally had. Alright, so, good on that?
Yeah.
That's the first 11 or 12 questions out of 25 is all this.
Wow.
Alright? So, yeah, wait. 12. 12 out of 25. Like this.
Okay.
Alright? Some of them are pictures, match the picture with the thing. Okay? Can I erase or not yet? Do you need a picture? Am I good? I'll stay with it. I'll stay. Yep.
Is this it?
So this is 12 out of the 25 questions. The others, and I'm going to do an epistasis,
Wait, 12 out of 25?
are on here right now.
That's crazy.
Because the whole FRQ part is all taught. So
Oh.
that's why if you count, if each of the multiple choice and FRQ are weighted equally, which they pretty much are, like 25 versus 24 points. So then 24 are like straight to the
Wait.
express probability, that stuff. So this, it's really 1/4 of
Does it ask, like, what phase is this? Or, like, what does anaphase happen? What happens in anaphase?
no, so we sleep it off, you like this picture and go, what's happening there, and like, what phase? Don't erase? Don't erase? Erase? Alright. What?
I haven't read this
Guys, I had so much coffee, like I had so much coffee. I forgot my water, so I drank all my coffee.
Like, remember it has to get a signal, and then it...
I'm sad. I know.
How much I will write that.
Can you send me pictures of your notes?
Yeah.
Because half of them are going to be on that stuff
I got it.
because...
Yeah.
Okay.
Okay. Next one.
I just saved you.
Thank you so much. I wrote down growth factor.
Wow, you have nice handwriting.
Thank you. Where is it? It's like I forgot It's like the capacity.
T is necessary for toes.
Do I need to know it?
I'll tell you about it. Yeah.
I haven't read chapter 7.
Alright.
Did it read tonight?
I'm gonna do your epistasis problem, but I've changed the scenario slightly. It's a little bit ridiculous, but it's okay because it represents. You'll get to the problem and you'll be like, oh, that kind of goes with that. Okay. T is necessary for toes. Okay. So toes are a dominant trait. Little t would mean no toes then, right? So first of all, your inference from this statement, big T, capital T is necessary for toes. So little t is no toes. There we go. Okay. Capital F, a dominant F allele. So again, like you have to read into the question. This is kind of how it's phrased, so that's why I want to interpret it for you. So this will be given to you, this information. I'm kind of telling you some additional information like that I'm interpreting by reading the question. So capital T, you need to get toes. Capital F results in no feet at all. Okay? I know, it's stupid, but it'll make sense when you see the problem. If capital F results in no feet, then if I don't have feet, do I care if I have toes or not? Like it doesn't, I know where to put them. And so of course it doesn't matter.
But if they just have the toes just come
So this is like the controlling gene or the one that has like sets a precedent over the other. Like F kind of, the F gene, like capital F little or lowercase f, kind of gets to dominate. And so again, if I end up with a capital F allele, like capital F or capital F lowercase f, I don't even need to refer to my T gene because I have nowhere to put my toes. Okay? So the question will be something like, if lowercase everything. Oh wait, no, I lied. It'll be lowercase. So little t, and then heterozygous for F. So big F, little f.
Wait, what's that?
Crosses.
This is
If.
What looks like a Y? Oh, this says if. Okay, I'll write it.
Oh, I was like, what is that?
If the, and it'll say it in words. If there is a cross between little t, big F, little f, and big T, little f, okay? Then what it's going to ask you is how many of them have blah, 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 okay? No toes or no feet or something like that. So let's do, well, we can do either of them. We'll just set it up and then we'll figure out them all out, okay? So this is good practice for two things, which is why I wanted to do it, and then this might have to be the last thing we do. I'm going to tell you a couple more things before we go, but then otherwise that'll be it. But this also covers the multi-trait, like how we do those, okay? I have two sets of letters. I have T's and F's. So remember, these, you're going to do two Punnett squares, all right? This will be my T1. So for my T Punnett square, I get that. I'll also have an F Punnett square. It actually kind of looks the same, right? Just with other letters. Okay. In each of these cases, though, look. I get 50% big T, little t, 50% little t, 50% big F, or sorry, 50% big F, little f, 50% little f, little f. So here are my combinations of like possible babies I can have, right? I can have big T, big F, little f. I could have big T, little f, little f, little t, big F, little f, and all littles. Okay? My probability for everything, remember, in this, like, it was 1/2 for big T, little t. It was also 1/2 for big F, little f. Since all of them are that way, my probabilities for all of these come out 1/4. Right? So the probabilities are equal that I'll get these 4 genotypes. Now, just briefly, because we don't have that much time, look at that and try to figure out, really quick, because this is part of your question. Knowing this now, figure out, label them. So some of them are going to have feet and toes, like everything looks like a normal thing. Some of them have no feet, right? And some of them have feet, but no toes. Okay, nobody, and here's the key, nobody has toes with no feet. Because how? And what are you talking about? Okay, so you can't have toes with no feet. So maybe the best way to go would be circle the ones, or one, that have no feet. First start with that. Remember that the controlling gene is F, so figure out which the oh my god, that's a weird one. Figure that one out, and circle the ones that I don't even want to look at the T gene, because why do I care? They don't even have feet. Do you see the ones who have no feet? Okay. So, capital F results in no feet. Yeah? Okay. So these guys with a capital F, if that results in no feet, then I do not care about their toes at all, because they have no feet. So I'm going to say 1/2.
None.
There's going to be one word.
Feet but no toes.
No feet, right? Now, I need to have a capital T if I'm going to have toes. So these guys got feet. They are very lucky. So they can walk. However, only this one has feet and toes. Okay? So only 1/4 of these people, or things, are lucky.
That's a sad one.
Isn't this depressing? So 1/4 of them have feet. They're happy about that. But they have no toes. I've heard it's hard to walk with no toes because it helps you balance. Even though, I don't know, they seem so short, but
You don't need them.
I feel like you don't, but then you can't wear flip-flops, so you do need them, because flip-flops, necessary. Okay, now, so this is pretty much number one. This is the epistasis problem. It just obviously isn't about feet and toes, because, yeah, whatever. But that's the same idea. So you go through it step by step. Do your 2 Punnets first. Figure out the dominant, figure out the one that's like the other one gene, and just circle whoever it applies to. Like, okay, you don't even, remember it's albinism was the example always. Like, if you don't get a fur color, then don't even look whether it's brown or black, all right? Super duper last thing, and then we have to go. Unless you want, do you have a question right now? Because I can do something. Okay. There was a question about crossing over, so I'm gonna, and there's one question on the test about it. Can I, I'll just erase this part, because you already knew that. Here we go. Okay. In an individual with big A, big B, big B, and blah, 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 okay, if we were doing that cross, okay? The gametes that we can create are capital A, capital B from that individual, and lowercase a. There's no other choice, right? However, what we would normally expect is that they're kind of mixing and matching, perhaps later in life. Okay. If though, I had an individual, so these are the possible gametes. There's a question about them. If I had an individual that's like that, we normally expect, right? We normally expect I could get gametes AB, 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 or AB, right? I have 4 choices. Sometimes though, if these are linked together, let's say this was grandma and grandpa, this is the P generation. If this is the F1 generation, which that should make sense, right? If I have, this is P, they have babies, and then look, isn't that what would be made, right? I mean, I would have A, B, B, right? Everyone's following that? Okay, good. Now, this individual should be able to make 4 types of gametes, but if I see something kind of weird with my numbers, it may be that these were actually on 1 chromosome. So I'm going to have AB and AB that were handed down to this person as 1 chromosome. Big A, big B. All right. now the likelihood has changed in terms of the gametes. If this is my chromosomes, or that's this person's chromosomes, then it is very unlikely, isn't it, that an A and a B would be inherited by my child, right? Because if this is the I'm going to pick, when I create gametes, I'm going to pick one of each letter to give to my child. But now new information, they're on the same chromosome, so it's more likely that I'm going to give capital A and capital B to my kid, right? Because they're linked. Does that part make sense? All right. However, remember that now I've replicated, the S phase has happened, I've replicated my chromosomes. And now during prophase 1, I might have some crossing over. Okay? If I have crossing over, that means I'm probably, because crossing over doesn't happen 100% of the time, I have probably more of those and more of those that were like grandma and grandpa, okay? If my original genotype was like the recessives were together and the dominants were together, in order to have this swap, I'm going to have crossing over, right? For my child to inherit a capital A and a lowercase b, there would have had to be crossing over kind of in between here. Does that part make sense? Okay. If we see numbers that seem kind of weird, like there's a lot that look like grandma and grandpa's gametes, then probably they're on the same chromosome, okay? The kind of funny thing, and this is why I'm telling it to you now, oh, and then we have to go, is I could give you numbers, I'm going to give you numbers, and I'm going to tell you how many there are of each one, and it tells us the distance between the genes, so I'm going to make it easy on us, okay? Let's say I have 35 of each of those, like if I could count them up. I'm going to have 35 individuals that inherit little a, little b, 35 that inherit big A, big B, and I'm only going to have 15 of each of those. We good? All right. What we do is we do the recombinants, like the switched divided by the total, which in this case, 35 plus 35 plus 15 plus 15, I made it 100 on purpose. All right, good. So 30 over 100 is like 30%, right? Good. Okay. This is in the book. There's only one question. If you're like, huh? Here's my hint. Take the littler numbers and add them together, because that's going to be the recombinants. It's always a lower number. Whatever the littler numbers is, add them together, divide by the total. Okay? It tells you the percent that have switched places. Okay? Normally, it's kind of weird to switch places, but some will. So we take the number that have recombined divided by the total number, and then that gives you a percentage, and then that's it. Okay? Good. Okay, now we have to go because my son has to go to basketball and get on time, and then he's not at me, and then he takes me.